Alright, welcome back everyone, ladies and gentlemen. We have a lot of material to get through today, so let's just jump right into it. So, if you see on the screen here, we have DNA replication. So, what is DNA replication? DNA replication is synthesizing new DNA through a process uh, of similar to transcription. It's going to read the DNA sequence, but instead of synthesizing mRNA, it's going to synthesize DNA. And it's how you, it's usually done if you see here with this box. We have G1 phase, which is like a checkpoint of growth, and then you have synthesis, the S phase, synthesis phase. That is where DNA is synthesized. So that's what we're going to focus on today. Then there's G2 phase, which is another growth stage and a final checkpoint before you initiate mitosis. I'm thinking mitosis is going to be the next video, and then we'll go into Cancer 101, and then we can start. Or actually, no. Mitosis, meiosis, then Cancer 101, and then we'll start reading these articles about the cutting edge science. So we're almost there. We're almost to the fun part. But before we get to the fun part, we got to do the unfun part. So, as I mentioned earlier, DNA replication is a semi-conservative model, meaning that it's somewhat conserved. So you have DNA, it's split through here. You break those hydrogen bonds between the nucleotides, the guanine and the cytosine, and the thymine and the adenine, and it splits. And this parental strand goes over here. This parental strand goes over here. Notice that 3 to 5 and 5 to 3, 3 to 5 and 5 to 3. Those details will be important later. And the newly synthesized strands are complementary to the parental strands, and they can be different, but that's why it's semi-conservative, because the original template, the original parental strand, is only halfway conserved in both new strands. Excuse me. So, first thing, DNA is semi-conserved. Oops, let's move the track. Okay. Semi-conservative. Okay. Next, DNA replication must occur in the 5' phosphate to the 3' hydroxyl direction. The phosphate of a new nucleotide must be added to the 3-hydroxyl side, the OH. Just put that, we'll make this abundantly clear. There we go. Hydroxyl side of the preceding nucleotide. Next, so semi-conservative, 5 to 3 direction, and DNA replication is bidirectional, meaning it happens in, you guessed it, 2 directions. So we have the leading strand and we have the lagging strand. So we have the initial 2 parent strands, which you see are spread, and then those arrows represent the leading and lagging strand of DNA. So you got the lagging strand. I'll just draw an arrow to it. So, boom. Okay, so you see here we have the leading strand, which is this newly synthesized DNA. So let me start from the square one. So we have the DNA here, right? This is the DNA. This is something called the origin of replication. And then you have the leading strand. The leading strand goes, it's the 3' to 5' direction, and it moves towards the replication fork. This is the replication fork, as I've marked here. So the replication fork is the direction that DNA is being synthesized. So it's being opened, and the strand is following the DNA strand as it's opened. And then the other strand, the one going in the other direction, the lagging strand, the parental strand for that is 5 to 3. The lagging strand is going 5 to 3 in the opposite direction. So you kind of see how it has to work, right? If the parental strand is 3 to 5 and it's moving in that direction, then the leading strand has to go towards that, because it has to synthesize DNA from 5 to 3, and it's anti-parallel. So it's going to move that way, and the other one is just going to be the opposite. Um, so that's a really brief synopsis. Now we're going to do so DNA replication, like transcription, like translation, has an initiation, elongation, and termination process. And initiation starts with the origin of replication. So what is the origin of replication? The origin of replication is a base nucleotide sequence that is AT rich, so adenine and thymine rich, and it's because they have hydrogen bonds. They only have two, so it takes less energy to break two hydrogen bonds than three hydrogen bonds. With guanine and cytosine, we know if you looked at the other video, apple tree, golf cart has more. That's just how I remember it, but three hydrogen bonds. It's going to take a lot more energy to break three bonds than two bonds. So the origin of replication, you just remember it has adenine and thymine rich area or region. And it's important to know that eukaryotic cells have multiple origins of replication. So there could be DNA up here and DNA down here, and they could both be considered origins of replication and start replicating the DNA separately. So how does this initiation process start? So there's something called the pre-replication protein complex, which binds to these adenine and thymine rich origins of replication, and it separates the DNA there. Creates like a little bubble for the next protein. Well, there's going to be a couple proteins, but it creates a bubble. So it breaks the DNA, so it's not together. So it severs those, cleaves those hydrogen bonds, and allows something called the single-stranded binding protein to bind the DNA, because those hydrogen bonds, if you know anything about hydrogen bond characteristics, they're going to want to come back together. So, and that process is called reannealing, when the hydrogen bonds want to come back together. So they prevent the single-stranded binding protein prevents the parental strands from reannealing to one another, because nucleases, enzymes in the nucleus that want to break down that or degrade that single-strand DNA, because it's not DNA. DNA is double-stranded, as we know. But nucleases want to degrade that short single-strand DNA phosphodiester bond. So single-strand binding protein binds to it and kind of just holds it there. It's like, nah, you're not gonna get back with that DNA bond. You're not gonna hydrogen bond back together parental strand. We're not having that. So that happens, and then helicase, which I've marked here, continues to unwind the DNA in this direction. So we know if this is the parental strand, this one right here, then the leading strand will be synthesized following that helicase. So what is helicase? Helicase unwinds DNA by cleaving hydrogen bonds that connect parental DNA. So after the pre-replication protein complex binds and severs those initial hydrogen bonds, helicase starts doing it, but it does it a lot faster, and it requires a lot of energy to do that. So helicase unwinds DNA by cleaving hydrogen bonds that connect parental DNA, and requires significant amounts of ATP to function. And this causes something called supercoiling. So have you ever tried to untie a string that was tied together? Like when you start separating and you pull those sides apart, the rest of it gets coiled or kinked up, right? So it gets really messy. It just starts super coiling, and it makes it so that this helicase can't go forward. The supercoiling stops the helicase from advancing. So what is going to deal with that? There's an enzyme called topoisomerase. Topoisomerase prevents supercoiling by making cuts with nucleases, those nucleases that like to, you know, sever those phosphodiester bonds, and it uses a nuclease domain. So the topoisomerase uses the nuclease domain to sever that supercoiled area and loosen it up a bit. And then it has a ligase domain. So they have two domains. This protein or enzyme has two domains. One domain that cuts and cleaves to loosen the supercoils, and one that binds them or reanneals those strands. So the ligase domain. And ligase is an enzyme that connects two molecules together. That's the gist of ligase. So topoisomerase cuts the supercoils and then stitches them back together when they're not as tight. So eukaryotes use topoisomerase 1, type 1, and this doesn't require ATP, which is good. It's not very energy taxing because we know that helicase is very energy taxing. So it doesn't sound very efficient to have two, energy taxing enzymes working so close together. So then you have topoisomerase type 2. This requires ATP and can insert negative supercoils. So, I'm saying I'm a lot. Anyways, topoisomerase type 2 inserts negative supercoils, which utilizes ATP to almost prevent supercoiling in the future. So instead of topoisomerase 1 that just goes along and just finds a supercoil, cuts it, and then reanneals it, topoisomerase 2 is like proactively, and it's taking preemptive measures to prevent supercoiling. And in eukaryotes, they just use type 2 and type 4. Type 4, similarly to type 2, requires ATP and can insert negative supercoils. So, excuse me. Prokaryote topoisomerase both utilize ATP and can insert negative supercoils. Eukaryote, they have both topoisomerase 1 and topoisomerase 2, one that doesn't require ATP and one that does and can insert the negative supercoils. So, clinically, topoisomerase type 1 can be targeted with irinotecan, topotecan, which I believe are topoisomerase inhibitors. So that can prevent this replication process. Topoisomerase type 2 can be targeted with toposide and tenoposide, which are, again, topoisomerase inhibitors. And that's clinically significant because they're therapeutic targets, since type 2 and type 4 are in prokaryotes, but eukaryotes can always use type 1. And then you have type 2 inhibitors, fluoroquinolones, or moxifloxacin, and this increased the nucleus domain, so it just cuts up more and it inhibits the reannealing or ligase domain, and this prevents cancer bacterial cells from replicating because bacteria uses that. I think, they use those prokaryotic topoisomerases. Not too sure. I believe bacteria is prokaryotic. I could be wrong there. Yeah, my bad. But cancer cells have a way of uncontrolled replication, right? So, inhibiting something that's in charge of loosening up those supercoils is a potential therapeutic target. So that's initiation. So after the replicative protein complex binds, the helicase, or the single-stranded binding protein, holds it there. Helicase pushes forward and replication has begun. So what is the replication process? This is elongation. So earlier I mentioned there was a leading strand. So there's parental strands, the original DNA. There's a leading strand and a lagging strand. The leading and the lagging strand are both the newly synthesized DNA. The parental strand is, of course, the OG, the original. Um, so the leading strand, the take home here is that the leading strand replicates towards the replication fork. This is important. Um, so it follows the helicase. This helicase is trailblazing through the DNA strand. The leading strand of newly synthesized DNA is being synthesized right behind it. So how does this happen? An enzyme called DNA primase starts this process. So if we have the DNA here, so here's the leading strand. I'll just outline it in blue so we can all stay on the same page here. And it's reading these nucleotides. So it's got a T. Primase has to go first because polymerase 3 needs a 3' hydroxyl group to add on nucleotides. It cannot add on nucleotides unless there's a 3' end. That is imperative that you understand that. Basically throughout replication. So what primase does is like, okay, that's the only thing we need. I'm going to stick on the first RNA primer, not DNA, and get you started. So DNA primase, which is here, as we see, the 3', 5' on the right is the leading strand, or the parental strand. So the leading strand is going to be, I can't really fit that in there. Sure I can. Never say you can't. There it is. Well, anyways, so the leading strand goes that way, and the primase is going to start by synthesizing the first nucleotide here. It's important to know that primase lays down RNA primers, so they're RNA, first of all. Second, they enable DNA polymerase type 3 to synthesize the DNA in the 5 to 3 method. And RNA reads the DNA from the 3' to the 5' and synthesizes the RNA primer from the 5' to the 3' end on the leading strand. So after that initial primase adds that first RNA primer to the first nucleotide at the origin of replication, you're going to have DNA polymerase 3. What is DNA polymerase 3? DNA polymerase 3 needs 3' OH of RNA primer, and it reads the DNA from 3' to 5'. So like I said, it's just going to read this as helicase unwinds this DNA strand moving to the right. Or I don't know if it's the same on that, but from the 3' to the 5' end of the parental strand, it's going to synthesize from 5 to 3, the leading strand. And what it's going to do, it's going to take that initial primer nucleotide, RNA primer, synthesize a nucleotide, a complementary one, from the 5' to 3', and just add and add and add and add until it finishes when the helicase reaches the termination sequence. But we'll get to that later. So that is DNA polymerase 3. The note here is that it's continuous and there's only one primer added at the very beginning of the leading strand. This RNA or DNA polymerase 3 also has a proofreading function which utilizes 3' to 5' exonuclease to replace incorrect nucleotides in the parental strand. So while it's adding these nucleotides to the leading strand, after reading the parental strand, it's just adding them. It's also reading that parental strand and being like, okay, is this the correct one? All right, boom. Is this the right one? Boom. If it's not the right one, it's like, nah. And then puts the right one and then incorporates the right nucleotide. So it has a preliminary proofreading function, essentially, which is important. So now we're going to move to the lagging strand. Now this is the fun strand. So the lagging strand, if this top if this newly synthesized um, nucleotide sequence is the leading strand, then the one going in the opposite direction is going to be the lagging strand. But the lagging strand looks more like this. It's broken up. And I'll show you why in a second. This is going to be the tricky part of this lecture, or should I? Because it's not going to be fun, but it'll be fun. But it'll be interesting to see how we follow along here. So the lagging strand contains multiple RNA primers and DNA stretching. So wait, it contains both RNA and DNA? Yes. Now why is that? So as we know, helicase is moving this way, but the RNA lagging strand is being synthesized this way. As you can see, the 3' end is over here and the 5' end is over here, which means these nucleotides are being synthesized 3,5, 3,5, 3,5, hydroxyl phosphate, hydroxyl phosphate, hydroxyl phosphate. And we know that DNA can only be synthesized 5' to 3'. So now here we have a problem. We're trying to elongate it this way, but we can only move this way. So how are we going to get through this? Well, let me explain. Primase, remember that enzyme from the first primer that adds the first nucleotide to the leading strand? Well, it's going to come back, and it's going to synthesize an RNA here and an RNA here. Let's say that. And I'll show you why in a second. We'll get to that. That's part of the termination, which is what I'll explain later. So the RNA primer comes in and adds a complementary T, as we see the A, and then it synthesizes a complementary T again. Cool. For continuity. Then, after the primase lays down those primers in the accessible nucleotide sequence, because helicase is moving this way, so primase is limited to what it can bind and add those RNA primers to based on where helicase is. So after the primers are added, DNA polymerase 3, same one as the in the leading strand, but a different enzyme, of course. It's probably can't do both at the same time. Is gonna elongate it this way. It's gonna add, because we know that the 3' end is gonna be over here on this side, complementary, anti-parallel to the 5. And the 5' is going to be added complementary to the 3', like that. So it's going to add this and move that way, move to the left. So again, you can just duplicate this. Yeah, I'm getting better with this software, man, I'm telling you. So 5', boom. What does DNA polymerase 3 need to elongate? A 3' hydroxyl group. So, see this? Here's where the magic happens. I'm going to use a different color. So, use red. So it's going to see that. DNA polymerase 3 is going to see that 3', add a nucleotide. Boom. Now it's going to see that. It's going to, and that is basically the extent of that. It's not going to connect there. That's a detail. That's important. So it adds here. In actuality, it probably just looks like this, and it connects to the preceding one, to the one in front of it. And that's pretty much what DNA polymerase type 3 can do. Following that, DNA polymerase 1. So here's the difference. The difference is DNA polymerase type 1 removes the RNA primers via the 5' 3' 5' to 3' direction exonuclease activity. It excises those primers. So you see, we had RNA primase add these baby blue primers, but we don't want primers. We want to replicate DNA. So it's going to remove those. It's going to, hey, we don't want you. And that is the major difference between DNA polymerase 3 and DNA polymerase 1. DNA polymerase type 3 just synthesizes a continuous nucleotide sequence. But DNA polymerase type 1 goes over this, and every time it runs into a primer, it removes it, adds a nucleotide. Adds it, adds a nucleotide. But the same deal. It synthesizes from 5' to 3' and it needs a 3' hydroxyl group to add. Yeah. The main difference is that it can excise RNA primers. So if you have all these primers mixed in with the DNA and you remove all those primers, now you have a bunch of DNA strands, but they're not connected, right? They're not connected because you can't just, if you excise something, it's fragmented. So that is called the Okazaki fragment. When you have a bunch of DNA on the lagging strand, the newly synthesized DNA on the lagging strand is non-continuous because DNA polymerase 1 was excising all of those primers, making the strand non-continuous. One way to inhibit this replication process is to use nucleoside reverse transcriptase inhibitors, such as didanosine, which is like basically di-deoxyribonucleic acid nucleotides. Basically, they don't have either hydroxyl groups. So on the, I think it's the 2 and the 3. I could be wrong, but the 2 and the 3, second and the third carbon, are missing the hydroxyl groups that are usually there. And so instead of adding, a DNA nucleotide with a 3' group on it, like see here how it has the 3', it's going to add one that only has the 5' and no hydroxyl group. Therefore, DNA polymerase cannot bind and cannot synthesize more or elongate the newly synthesized DNA sequence. But that's just a nugget of, if you wanted to know, kind of stuff. So then after all the DNA polymerase type 1 is done, excising all those DNA fragments and replacing those RNA primers with DNA, newly synthesized DNA, ligase comes along and just stitches them all together. Ligase basically goes, oh, I shouldn't have removed all those. Ligase goes, yeah, and stitches those bad boys all together. I think I'm about to run on time because I know that this thing only lets me record for 30 minutes. So I'm just going to go ahead and pause it. All right, welcome back. So if we remember correctly, we just got through connecting the newly synthesized DNA. Remember, we excised those RNA primers using DNA polymerase 1, and ligase just came and stitched them all together, synthesizing our newly synthesized DNA, specifically the lagging strand, because it's Okazaki fragments that are fragmented DNA on the lagging strand. You have to stitch them all together with ligase. The leading strand didn't have that problem. All you had was DNA polymerase knock out that first primer, and that was it. So there's no cutting up, stitching together. There's one cut, replace, and it's seamless. On the other one, it's cut, stitch, cut, stitch, cut, stitch. So it's just cut, 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 replace, stitch them all with ligase. Um, so that's the difference. Um, so now we're going to move on to termination. So when does this process stop? So termination occurs when helicases, those enzymes that were opening up and severing those, this guy, the guy leading the way, um, when they meet each other. So say this guy's going this way, this guy's going this way. Ooh, those arrows are tough. Ooh, ooh. Excuse me. That's horrible. Anyways, when they meet, they send signals and they just go, oh, okay, I guess we're done. And then they meet in the middle here. They say, hey, what's going on? Ain't nothing much, man. And then they bump each other off. And that basically stops that replication process because, the polymerases are going to meet and then they're just done. So that's basically when you know that replication of those strands are complete. Excuse me. So what happens at the end of these DNA strands? I don't know how familiar you are with telomeres, but telomeres are like the cap on DNA. So they basically keep the helix, tight. They're like caps. They're non-coding DNA. They don't code for RNA. And they prevent gene loss by buffering the DNA shortening process of every subsequent replication cycle. So the thing about replication, which is it's kind of cool because like when I think of it, it's like Wolverine if you're into that stuff. Um, the regenerative property, or Deadpool. Um, we got limits, unfortunately, and that limit comes from telomere and telomere shortening. So at the end of the genomic sequence, you have these telomeres. And every time you replicate, they get shorter. So if this were the telomere, these were the telomeres. Every time you replicate, they get shorter and shorter until they're gone. And once they're gone, that's called the Hayflick limit. That's the number of replications that can be done before replication starts to impact the actual coding DNA, which is in here, your genes, your eye color, other things that are coded by genes. I'm not going to get into specifics here. And that's not good because then it has actual consequences on like protein synthesis. And I bet there's a number of diseases that can come from that, result from that. So how does this happen? Now, this is going to be the most fun part of this lecture because basically it's going to be an application of everything that we've learned thus far. So as we know, the leading strand, we're going to use our colors for this, has those RNA primers, right? It has one RNA primer. Use the right color here. And DNA polymerase 1 removes that primer, smooths it together, makes it seamless with the rest of the newly synthesized leading strand, right? So remove the one primer and you're good. But So we're still assuming that helicase is moving this way, but it ran to the end of the line and it fell off. So helicase done gone away. So now DNA polymerase 1 specifically goes down the line. Goes down the line and removes these. It removes these strands, right? It takes this. It's like, nope, nope, we don't need you. You're an RNA. We need DNA. And it puts the DNA. So I use dark blue for DNA, just because we needed some different colors here to really illustrate what's going on. So then replaces that RNA with DNA, and those nucleotides then get synthesized together by ligase. For a seamless DNA strand, we're going to use black so that we know where it's seamless. So boom, good, we're cooking. All right. DNA polymerase 3 already elongated from the other, or it already elongated it, right? Because it's going through and it elongated from the previous primer, right? So you see that hydroxyl, that 3' from the RNA, that's the turquoise color, right? So that's an RNA primer. The DNA polymerase, sees that elongated, right? So now we're out of space, right? DNA polymerase 1, which plucks off, oops, that's not right, plucks up the RNA primer portion at the end of the DNA, right? So this is the end of the DNA. When DNA polymerase 1 plucks off this RNA primer, it got rid of the RNA primer and everything involved with it. So when it removes the primer, it removes those nucleotides. And when it removes those nucleotides, it removes the phosphates, the hydroxyls associated with those. So now we have a 5' end and essentially the end of the line, the end of the DNA strand. There is no 3' for which you can add the nucleotide and finish off the Okazaki fragments of the lagging strand. That's a problem. So instead of, we're not advanced enough we haven't evolved to deal with this in regular cells. We have evolved to deal with this in stem cells, which I will talk about later. But in your run-of-the-mill cell, that's it. So you're done. That part's just lost. You just lose that part of the telomere. Because this is at the end of the DNA strand. What's at the end of the DNA strand? Telomeres, right? So you lose that part of the telomere. So what happens? It gets shorter. Not by a lot, but by enough. Do that over and over again, you start seeing the impact. So that limit is called the Hayflick limit. Once this process of telomere shortening starts to actually impact coding DNA. Okay, so now let's talk about the cool stuff, because this is actually some research I wanted to get into next. So I think the first journal paper that I want to talk about after we do mitosis, meiosis, and the replication cycle um, is going to be dealing with telomerase. Because I remember one of my colleagues gave a talk about telomere shortening, and I thought it was fascinating at the time, but I was researching something different. I was doing breast cancer research at the time. Um, but she gave this talk about telomere shortening, and I remember that being like really cool and how telomerase kind of elongates that and how that can be utilized in stem cells, and it was really cool. So we'll probably do something with that. But what is telomerase? It's an enzyme that deals with telomeres. No, I'm just playing. Uh, Telomerase is a ribonucleoprotein that uses reverse transcriptase to generate DNA from RNA. One thing, just side note here, is whenever you see reverse transcriptase, think RNA to DNA. That's just got to be like automatic. Because transcription is DNA to RNA. So reverse transcription is just RNA to DNA. So you're generating DNA, which is unique. Because how are you going to, I describe DNA as the secret Krabby Patty formula. How are you going to generate the secret Krabby Patty formula from a copy? Doesn't make sense, but it does. Because telomerase can do it. Anyways, we're staying focused here. Basically, telomerase is an enzyme that uses RNA nucleotide sequences, specifically this one, AAUCCC, to synthesize TTAGGG. So it uses adenine, adenine, uracil, cytosine, 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 the RNA nucleotide sequence, to synthesize thymine, thymine, adenine, guanine, guanine, guanine on the parental DNA strand, thus elongating the telomere and preventing telomere shortening. I'm going to say that again. So it synthesizes, the enzyme telomerase uses RNA, specifically the sequence adenine, adenine, uracil, cytosine, 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 to synthesize parental strand DNA, specifically the non-coding region TTAGGG, and elongate the telomere of the parental strand. This prevents telomere shortening, and this works because telomeres do not code for RNA. Telomerase is common in stem cells, neural stem cells, and neuroprogenitor cells, and can be mutated in some cancer cells, which is why they're allowed to proliferate uncontrolled. They're capable of uncontrolled proliferation. So they just keep replicating. You think they die from telomere shortening, but they don't. And it's because some of them have found a way to harness the power of telomerase to basically activate Deadpool powers and basically be, incapable of death. Yeah, so that's pretty cool. We'll talk about that in another video. Sorry this one was so long. I tried to go through it pretty fast, but if you have any questions, as always, leave them down in the comments, and I'll address them to the best of my ability, and hopefully we can learn something together. Till next time, thank you.